Hi everybody, I hope you all are doing great, and we're almost at the end of the semester, so there's just a little bit more to do. I thought that this would be helpful to talk a little bit about what's required for Lab 9. I have extended the date one more day, so you have until 11 p.m. Tomorrow to complete this lab. All right. Okay, so this is all about transcription and translation, which is what you've been learning about in these modules. So, of course, transcription is going to occur by an RNA polymerase. There are at least 3 RNA polymerases that you read about in that chapter in the module. So, first you're given the gene, and the gene is there, and it is going to make a protein. In this case, it's going to make a protein, and that protein is going to affect the color of the coat of the rock pocket mouse, the very cute little mice. So this is just a portion of the gene, and of course this is just one part of the table. For table 1, this is for the normal. A normal gene is called the wild type gene, and then if there's a mutation, you compare it to the wild type, which is going to be a change in the DNA sequence, because that's what mutations are all about. So you're given the gene, and you can see that they've split it up for you. Of course, in the gene and on the chromosome is just a sequence of bases. There's no separation between one group of 3 and the next. So that happens on the ribosome, but you're just given the sequence, and we're going to split it into blocks of 3, because that's how the ribosome is going to read it. It's going to read it as a codon. It cannot read DNA. The ribosome cannot read DNA. It must read messenger RNA for it to take that information and translate it into an amino acid. That happens on the ribosome, and of course it's going to translate each one of these codons to an amino acid, unless it is a stop codon, and then that's like the period at the end of the sentence. There's no actual amino acid which will be placed there. It's like the end. So when the RNA polymerase looks at the DNA gene, it can't just copy it directly. So it's not going to, well, first of all, there are no T's in RNA bases, so you need to remember that. There's a U takes the place of a T. But it can't just say TTG. That's not what it does. There has to be a hydrogen bond formed between the copy of the gene and the messenger RNA. So to form that hydrogen bond, you have to have complementary base pairs. So you're going to form the complement. So that would be AAC. And that, of course, is going to be an RNA nucleotide, not a DNA nucleotide, and so forth and so forth. And once you have that messenger RNA codon, now the ribosome, when it's running along the messenger RNA and reading it, it can translate that, because there's going to be a tRNA, which is going to be complementary to that mRNA, and on the other end of that transfer RNA is going to carry the appropriate amino acid. So that's kind of what you're doing. So here we have, so let me just stop this real quick. All right, so if we have a sequence here, and I've got TTG, and I have to write it in RNA, then I would write AAC. And an A will hydrogen bond with a T. Even though it's an RNA nucleotide, it'll still hydrogen bond with a DNA nucleotide. An A will hydrogen bond with a T, and then a C will hydrogen bond with a G. And then here, there are no T's in RNA. So you must put a U, and then of course a C and a C. And then I'll do one more, an A and a C and a C. So you can see these are complement, and hydrogen bonds temporarily form between the messenger RNA and keep it associated with the DNA gene. Once the transcription is done, everything falls off, because they're just simply hydrogen bonds. They don't stay very connected very long. And now you've got a messenger RNA transcript, which will be modified in the nucleus to remove the introns, add on a 5' cap, and a 3' poly A tail, and then that gets transported to the cytoplasm, and the cytoplasm is where this messenger RNA is going to be read by the ribosome and then translated into the appropriate amino acid. All right. All right, so if we carry on here, now how do you take these, the messenger RNA codon, and how do you, on earth, do you read this table? The innermost circle is going to be the first letter. The second circle outside of the innermost circle is going to be the second letter. And then the third circle farthest away from the center is going to be the third letter. So if we go back to this would be the first letter, so it's read left to right, 1, 2, 3, so A, C. So if we go to the table and we look for A, there is A, and there's an A for the second letter, and then a C for the third letter. So that first codon is going to be, you can either write out the full word, asparagine, that's a specific amino acid, or the 3-letter code ASN, and that's what you would put in the table. I've given you a different format of the table here. So